Hello and welcome to Technology Today for August 3rd, 2025. It's Season 2, Episode 31. I am MayorWatt. I run ohmTown.com. That's the site you see right there on the screen. If you've never been there and you're downloading the podcast, welcome to ohmTown. Go over, sign up, sign in, all news, no noise, because all the noise comes from me. So the way that this works is I aggregate a bunch of different sources into ohmTown. I have varied interests, pretty broad spectrum. There's 50 channels, which represent 50 shows on ohmTown. And, well, today we're going to be talking about the inner lives of cells, the Electron Tunnel, a Bronze Age warrior 6 feet tall, green tech environmental crisis, and a spectrometer on your phone. Pretty cool stuff. I'm a big fan of tech, science, and society. I talk about those three from the perspective of them meeting in the center of ohmTown, ohmTown.com. So I talk both publicly and here on the stream privately as from the perspective of science, technology, and society, STS. In academia, that's what it's referred to as typically, STS. And I love that convergence. You cannot separate all three of those individually. You can't even talk about one without the other two being impacted. So always talking about all three. That said, let's get into it. All right, the very first article, this is the front page of ohmTown, but this is why you're here, Technology Today. And the first article is these super resolution microscopes are revealing the inner lives of cells. Advanced light microscopy techniques are giving scientists a new understanding of human biology and what goes wrong in diseases. When you click on, see, we only aggregate this little snippet. We don't screen scrape. It's all provided by the sources, but we even limit what the sources provide. We limit it to a very short sentence or a small paragraph. There's only one in the entire cluster of everything that we aggregate, or I aggregate into ohmTown. That goes beyond our limits, and no matter what I do, they don't turn off the full flood. But anyway, when you click on the little visit the source, you get taken to our source. In this case, it's Smithsonian Magazine, smithsonianmag.com. And Katarina Zimmer from Knowable Magazine put this article together, but there's a graphic here on the front that says new light microscopy techniques can distinguish objects down to less than 10 nanometers. I think these are amazing pieces of technology. I actually launched a YouTube channel a long time ago, the Microscope channel, and was streaming real-time microscopy with live Petri dishes, basically. And but there was a real problem with it, and that is that it was all conventional lighting, and so the lighting would overheat samples and stuff like that, and so I didn't quite feel right with that, and so I shut it down. But using a tiny spherical glass lens sandwiched between two brass plates, the 17th century Dutch microscopist Antony van Leeuwenhoek was the first to officially describe red blood cells and sperm cells in human tissues and observe animalcules, bacteria, and protists in the water of a lake. You can actually look online on YouTube for these original microscopes. It was quite fascinating to see how they were constructed. But by 1873, scientists realized that there was a limit to the level of detail, and when light passes through a lens, the light gets spread out through diffraction. This means that two objects can't be distinguished if they're less than roughly 250 nanometers or 250 billionth of a meter apart. Instead, they'll appear as a blur. That put the inner workings of cell structures off limits. Electron microscopy, which uses electron beams instead of light, offers higher resolution, but the resulting black and white images make it hard to tell proteins apart, and the method only works on dead cells. Now, however, optics engineers and physicists have developed sophisticated tricks to overcome the diffraction limitation of light microscopes, opening up the new world of detail. The super resolution light microscopy techniques can distinguish objects down to 100 nanometers and sometimes even less than 10 nanometers. Scientists attach a tiny colored fluorescent tag to individual proteins or bits of DNA, often in living cells where they can watch them in action. So that's basically what's going on. Single molecule localization microscopy, for instance, takes advantage of the fact that spots on an image are easier to localize with precision than they appear, or when they appear in isolation rather than clustered together. In regular light microscopy, you would have to change your focal plane and look deeper, but you only see this little sliver. Everything else is blurry. So they say another technique, stimulated emission depletion, scans the samples with lasers that are surrounded by a second donut-shaped ring of lasers that cancel out the fluorescent light around the area of interest, sharpening the microscope's resolution. And then a third method called structured illumination microscopy illuminates samples with a striped pattern of light, and these stripes interfere with the light emanating from the sample in ways that allow scientists to infer additional detail about the image, basically use techniques to reverse those lines. So, revising biology textbooks, the techniques have already revealed new cell structures. Scientists have discovered that neurons have a unique kind of scaffold called the membrane associated periodic skeleton, or MPS, that provides rigidity and form and helps to regulate the signals that pass from one neuron to the next. And actually, neurons move about trying to find other partners to match onto and transition information. The MPS is involved in almost all of the neuronal functions, says Columbia University neurobiologist Victor Macaron Palacios, who recently reported that colleagues that a particular protein, paralemin-1, is responsible for organizing the intricate structure of the MPS. You can actually see it. Specialized microscopy techniques have revealed that nerve cells have a special type of internal skeleton called the membrane-associated periodic skeleton. A well-ordered nano-architecture of the skeleton is regulated by a protein called paralemin-1, fluorescently labeled in this image. You can see the actual striations of this skeleton, and it's very regulated. It's pretty amazing. So we barely understand what we are looking at. Scientists also have been studying how organelles interact with one another. Cell biologist Jennifer Lippincott-Schwartz of the Howard Hughes Medical Institute's Janelia Research Campus in Virginia, for instance, is scrutinizing the structures that mitochondria, the powerhouse of the cells, use to dock onto protein-making endoplasmic reticulum, which supplies calcium and fats to mitochondria. Pretty amazing stuff. There is a lot more over at this article. You can get the article out of ohmTown if you're over on Twitch. Otherwise, it's embedded in the show notes over on YouTube and in the podcast when it drops. This here is a picture of a fibroblast cell nucleus. The DNA-binding histone protein called H2B is fluorescently labeled in yellow. The top half was imaged with conventional wide-field fluorescent microscopy, your standard light type style of microscopy with a fluorescent tag imparted into the protein. The bottom half was imaged with the super resolution microscopy, showing basically a higher resolution of this particular protein H2B within the cell nucleus. Pretty spectacular resolution drop. Increase. The difference is that the glow was because of standard light. You had to capture a whole lot more. But when you don't have to open up the lens so much, in a sense, you get higher resolution. So the packaging determines how accessible a given region of DNA is, and that's important because different cells in the body, say heart cells or neurons, use only a specific subset of their genes. The ones that they use are left in a looser, more accessible state, while the silenced ones are packed tight. So pretty amazing. We'll be able to see more, be able to address more in a more focused way, and be able to count things for targeted therapies. Pretty amazing. I love this technology. Now I got to look into it to see how much this stuff costs. All right, let's keep going. The next article is over in Technology Today as well. It's aggregated from a different source, but possibly. I haven't looked at it yet. All of these articles I've never looked at, but I have some experience with in some way, either the technology in one way or another, or the science behind it, or how society is impacted. And that's the three domains that I specialize in. So, a century-old quantum mystery solved. Scientists finally see inside the electron's tunnel. A team of physicists have experimentally uncovered what electrons do during quantum tunneling, revealing a surprising internal collision process that challenges long-held beliefs. Professor Dong-Eon Kim of PosTech's Department of Physics, in collaboration with Max Planck Korea PosTech Initiative, has made a major breakthrough in understanding a fundamental concept of quantum mechanics known as tunneling. When you click the link over at ohmTown, it takes you over to the source. Pohang University of Science and Technology, or PosTech, put the article together. It's posted at scitechdaily.com. And it says the team of physicists have experimentally uncovered what electrons do during quantum tunneling. Although it may sound like science fiction, particles in the quantum world can actually perform feats that resemble teleportation. One such phenomenon, called quantum tunneling, where electrons manage to pass through energy barriers, often compared to walls that they do not have enough energy to overcome, as if carving a hidden path straight through. This effect plays a critical role in how semiconductors function, vital components found in smartphones, computers, and let's not dumb down the article too much. This is Technology Today, after all. While scientists have long known what happens before and after tunneling takes place, the exact behavior of electrons as they move through this barrier itself remained a persistent mystery. So the results revealed a surprising phenomenon. Electrons do not simply pass through the barrier, but collide again with the atomic nucleus inside the tunnel. The research team named the process under the barrier recollision. Until now, it was believed that the electrons could only interact with the nucleus after exiting the tunnel, but this study confirmed for the first time that such interaction can occur inside the tunnel. Even more intriguingly, during the process, electrons gain energy inside the barrier and collide again with the nucleus, thereby strengthening what is known as Freeman resonance. This ionization often, it says, was significantly greater than that observed in previously known ionization processes and was hardly affected by changes in laser intensity. That is a completely new discovery that could not be predicted by existing theories. So the research is significant as it's the first time in the world to elucidate the dynamics of electrons during tunneling. I don't think they actually witnessed it, though. They just know that it hits something and goes to the other side more energized than what went in. Through the study, we were able to find clues about how electrons behave when they pass through the atomic wall. Now we can finally understand tunneling more deeply and control it as we wish. There's more in this article, but we're going to move on. The next article is over in Wanted. Archaeologists unearth a 6-foot-tall Bronze Age warrior clutching a spear. A man who died roughly 3,800... Oh, it's not like a bronze statue. It's a actual person. They died 3,800 years ago, may have been a prominent military leader, and was buried in an elaborate fashion. Again, you click the link, it takes you over to the source. This one is Gizmodo. Natalia Mesa put the article together, gizmodo.com. There's a picture of this Bronze Age hero, 3,800 years old. It's pretty amazing. They say that the Bronze Age man was buried alongside precious artifacts in a massive tomb. The man's skeleton was measured over 6 feet 6 inches, actually, in height, and dates back to the Middle Bronze Age, approximately 3,800 years ago. Scientists believe that they may have been a prominent military leader due to the way in which they were laid to rest. The discovery offers a glimpse into social and religious beliefs and rituals of Bronze Age societies. This was all translated from a statement from the Azerbaijan Ministry of Culture. The burial mound was found near, I think it's Karankal Plain, in an area locally known as Yovsandlidar, near Azerbaijan's western border, measuring more than 90 feet in diameter and over 6.6 feet in height. The mound contained 14 one-ton limestone slabs face to face just beneath its surface, along with a stone idol shaped like a bowl and a circular limestone seal. Known locally as kurgans, burial mounds like the one were ceremonial structures often built by nomadic cultures, offering valuable insights into ancient societies' rituals and beliefs about the afterlife. At the center of the mound was a chamber, 6 feet wide, 20 feet long, divided into 3 sections. One section contained the human skeleton, another was filled with ceramic vessels, and a third was completely empty. The empty space may symbolize the resting place for the deceased spirit, according to the statement, but nobody really knows. The 4-pronged bronze spearhead is an especially rare find, not just in Azerbaijan, but throughout the broader South Caucasus region. Alongside the spear, researchers uncovered a variety of other valuable items, including bronze ankle adornments, obsidian tools, paste beads, and inlaid ceramic pottery, suggesting the individual held significant prestige. Inside the ceramic vessels, they found the bones of cooked animals believed to be provisions for the afterlife. Quite fascinating. There's always more here. Go over and check it out. Follow the link through ohmTown. The next article is over in Technology Today as well. How the rise of green tech is feeding another environmental crisis. The world's race to decarbonize has led to the rise of electric cars, and with it, soaring demand for lithium, which is required for the batteries. Again, when you click this link, you'll get taken over to BBC. Ian Wells, I think, is the author that put it together over at bbc.com. Raquel Selena Rodriguez watches her step as she walks across the Vega de Tilopozo in Chile's Atacama salt flats. It's a wetland known for its groundwater springs, but the plain is now dry and cracked with holes she explains were once pools. Before, the Vega was all green. You couldn't see the animals through the grass. Now everything is dry, she gestures to some grazing llamas. For generations, her family raised sheep here. As the climate changed, the rain stopped falling. Less grass made it that much harder. But it worsened when they started taking the water, she explained. They are lithium companies. Beneath the salt flats of the Atacama Desert lie the world's largest reserves of lithium, a soft silvery white metal that is an essential component of batteries that power electric cars, laptops, solar energy storage, etc. In 2021, 95,000 tons of lithium was consumed globally. By 2024, it more than doubled to 205. By 2040, it's predicted to be more than 900,000 tons. Most of that increase will be driven by demand for electric car batteries, says the IEA, the International Energy Agency. And everything is basically going away because of it. Chile's government has framed the plans as part of the global fight against climate change and a source of state income. Mining companies predominantly extract lithium by pumping brine from beneath Chile's salt flats to evaporation pools on the surface, and the process extracts vast amounts of water in this already drought-prone region. So the lagoons are smaller now. We've seen a decrease in reproduction of flamingos. She said lithium mining impacts microorganisms that feed birds. Everything is connected, folks. This is something that I talk about all the time. I practice a thing called associative thinking, where the process of just thinking about something, you can link it to other things, and it's a daisy chain of impacts, kind of like a butterfly effect, where a decision here in the Atacama Desert could drain the ability to keep life in that area. It overheats as well, and it throws off the environmental controls that are in place by nature. And nature abhors a vacuum, so you're going to see changes in the environmental dynamics in the region, and those are going to impact the areas around the world. So the underground water from the Andes, rich in minerals, is very old, they say, and replenishes slowly. If we are extracting a lot of water and little is entering, there is little to recharge the Salar de Atacama, she explains. Damage to flora has also been found in some areas on property in the salt flats mined by the Chilean company SQM. Almost 1/3 of the native algarobo or carob trees are starting to die. And they started dying as early as 2013 due to the impacts of mining, according to a report published in 2022. So how do you mitigate it? Frankly, I don't think that corporations really care, and they're going to move that from one place. That the value of that material is more than what people care about regarding the environment. So that, again, that sociopathy, the fact that nobody care, well, not nobody, but the people in control don't care. They're going to make their money, and they're going to live in a region where they're not impacted by it. So again, they don't see it, they don't care about it. Even if they do see it, they're making too much money to care about it, because when you get filthy rich, you stop caring because there's no social controls on you. So it says we're doing several pilots to understand which one works better in order to increase production but reduce at least 50% of the current brine extraction. Yeah. But if it doesn't cost more to do the brine extraction, then it's the least costly, which means that the profit margin is wider, and they still charge, a lot for it. So yeah, nothing's going to change until the cost changes. It's not the fact that they need to care about the world that is going to impact them, because they don't. All right, let's keep moving. The next article is over in Technology Today. Tiny spectrometer measures light across broad spectrum and fits on a smartphone. Researchers have successfully demonstrated a spectrometer that is orders of magnitude smaller than current technologies and can accurately measure wavelengths of light from ultraviolet to near infrared. The technology makes it possible to create handheld microscopy devices and holds promise for the development of devices that incorporate an array of new sensors to serve as next-generation imaging spectrometers. Yeah, I need one of these because I have an experiment that I want to run. I do not. So the article is over at techexplore.com. It's written by Matt Shipman, North Carolina State University. Gabby Clark and Andrew Zenin edited this and or reviewed it. The paper is titled Single Pixel Spectrometer Based on a Bias Tunable Tandem Organic Photodetector, and it's published in the journal Device. Spectrometers are critical tools for helping us understand the chemical and physical properties of various materials based on how light changes when it interacts with those materials. Says Brendan O'Connor, corresponding author of a paper on the work and a professor of mechanical and aerospace engineering at North Carolina State University. They're used in applications that range from manufacturing to biomedical diagnostics. However, the smallest spectrometers on the market are still fairly bulky. True. The ones that I've been looking to get are some 2 feet wide by 1.5 feet long, or the other way around. I mean, they're like 2.5 feet wide and half a foot or a foot thick and 2.5 feet wide. So I don't, God bless America, I don't know what's going on here in my brain. Anyway, they're big. They're like 3.5 by 2.5 by 1 foot or more. And so it says we've created a spectrometer that operates quickly at low voltage and that is sensitive enough for a wide spectrum of light. Our demonstration prototype is only a few square millimeters in size, and it can fit on your phone. You can make it as small as a pixel if you wanted to. So if you rapidly apply a range of voltages to the photodetector and measure all of the wavelengths of light being captured at each voltage, you have enough data that a simple computational program can create an accurate signature of the light that is passing through or reflecting off of the target material. The range of voltages is less than 1 volt, and the entire process can take as little as a millisecond. My problem there is that I want not a simple computational program that can recreate an accurate signature of the light, but I want the signature of the light. It shouldn't be at certain levels, measure all of the wavelengths of light. I mean, there has to be interstitial levels of light between one level and another level, right? Like 451 nanometers, 452 nanometers. There could be something that is a massive spike in the spectroscopy, but you don't see it because the calculation from one range to the other range, I don't like that. I'd have to see it. In the proof of concept testing, the researchers found their pixel-sized spectrometer was as accurate as a conventional spectrometer and had sensitivity comparable to commercial photodetection devices. Now I'm really curious. I want to see the results and how they actually got it. So I'm going to have to hunt down this paper and take a gander at it. At any rate, that is it for today. So we're going to fire up the spectrometer, and that's going to reveal Technology Today over at ohmTown.com. Go over, sign up, sign in, become a citizen, and be sure to follow us on Twitch and YouTube. Download the podcast, tell a friend, leave a message, come and hang out. Monday through Friday, we do the shows, although my mayoral duties have kept me from being everywhere, everything, everywhere, all at once, unfortunately. But that's it. We are done for today's Technology Today. So see you soon. Bye-bye.